Hello viewers, this is Pokazim Klosana from Luzidrift Secondary School in Mount Fletcher, and welcome to the Eastern Cape Department of Education broadcast studio. Today's lesson will be on basic agricultural genetics, and we will be focusing on the monohybrid and dihybrid inheritances. So this is the question 4 of your paper 2 for NSC exams. Then in section B, it will cover 35 marks. Then in section A, it will cover only 15 marks. So the total is 50 marks. Now, we will start with the monohybrid inheritance, which is the genetic cross between two individuals, which involves a single characteristic, meaning this characteristic is controlled by only a single pair of a gene. For example, the color of an organism, the height of an organism, or even the shape of an organism. Now, we'll make an example of an illustration for a monohybrid cross. We'll use color as an example. Now, we have two parents. These parents are the two individuals that I said they are crossed when I was defining monohybrid inheritance. Let's make an example. We have two colors here. We have a brown color and a white color. A brown color is represented by a dominant allele. A dominant allele is a capital letter B. That is a dominant allele. And the recessive allele is the small letter b. The small letter b represents the recessive allele, which is white, okay? And then we have two parents here. The male is brown in color, and also the female is white in color, right? And then let's come to the genotype. The genotype of a male is heterozygous. When it is heterozygous, meaning the alleles are in different forms. One allele is a small letter, meaning it is a recessive allele. Another allele is a dominant allele because it is a capital letter. So the recessive allele is always masked by the effect of a dominant allele, meaning it cannot appear on the phenotype of an organism. The female is a recessive, a homozygous recessive, a small letter B and a small letter B, meaning they are both recessive. They are in a homozygous state, meaning the characteristic can appear on the phenotype of an organism. This is a homozygous genotype. The first one is a heterozygous genotype, sorry. Then now, we are given a statement here. Use a genetic diagram to determine the genotype and phenotype of an F1 generation. Of the F1 generation, if the two parents were crossed, that is, the F1 generation is the first filial generation, the first offspring generation. So we're going to use two methods here. We're going to use a genetic diagram. We're also going to use the Punnett square. So let's start with the genetic diagram to determine the F1 generation, the genotype, and also the phenotype of these organisms, that is the offspring. Now, we have the first parent generation. These are the first parent generation, P1. And then we have the phenotype. The phenotype of the male, we said it is brown. The female is white. We also have the genotype. The genotype, remember, it is heterozygous. Then the female is homozygous. Small letter and a small letter. And then when we are talking about the genetic cross, meaning there will be a reproduction. For a reproduction to occur, there must be a formation of a gametes, and they get formed after meiosis has occurred. So meiosis occurs, and then the gametes are formed. Now, each gamete will contain one allele from the genotype, meaning we'll have a gamete with a single allele, a dominant allele. Another gamete with a recessive allele, then crossed with the recessive allele and another recessive allele from the female after the gametes are formed. Now, a fertilization occurs. Fertilization occurs. Fertilization, which is the fusion of a female gamete and a male gamete, which gives us a heterozygous individual. Another one will also give us a heterozygous individual. The third one will give us a homozygous and also a homozygous. So these are the 4 possible offsprings that we have. Okay, now let's move to the Punnett square. We have another method that we'll use, which is the Punnett square. When we do the Punnett square, you need to draw a square. You need to draw a square. When you draw a square, you just draw a square. You just draw a square. You don't have to go through all the meiosis and the fit. This is the Punnett square for a monohybrid. You just go straight to the Punnett square. For a monohybrid cross, you draw a square, 2 lines down, 2 lines across. This is a correct Punnett square for a monohybrid inheritance. Then the first box, we only write gametes. Gametes in the first square. All right, now we are going to use the genotype we were given before, now as the gametes. So we have for the capital letter, that is the dominant allele. Remember, this is the genotype for a male. A capital letter B and a small letter b. Then the gametes for a female, recessive, and another recessive. Then coming to the fertilization part. Fertilization part, that is the fusion. We're gonna cross the capital letter B and the small letter b. This is the capital letter B that we are using. This is the small letter b that we are using. Then after, this is a capital letter B, we have crossed the 2, and a small letter b, that is heterozygous. The small letter b and another small letter b, meaning it is heterozygous. The capital letter B, the dominant allele, B from the male, is crossed with the small letter, that is the recessive allele from the female, which also gives us again the heterozygous individual. And lastly, we have the recessive allele and another recessive allele, which gives us a homozygous genotype. So this is the Punnett square for a monohybrid cross. Now the question, determine the ratio of the first filial generation. We're going to determine both the phenotypic and also the genotypic ratio. Let's go with the genotypic ratio first. Genotypic ratio is, let's check in terms of the genotype, which one is heterozygous, which one is homozygous. We have only 2 offspring that are heterozygous. That is again 2 dominant and the recessive. 2 dominant and the recessive equals to 2 homozygous. This is the genotypic ratio. This is the genotypic ratio. Then coming to the phenotypic ratio. Phenotypic ratio equals to, in terms of the phenotype, we're talking about the physical appearance of an organism. How many are brown? How many are white? 2 are brown, capital letter. The ones that are heterozygous are brown. Is brown equals to 2 that are white. This is a phenotypic ratio for the possible offspring, that is the first filial generation. We're now calculating the percentage. The second question, we're calculating the percentage of the white offspring. How many white offspring do we have from the Punnett square? We only have 2 white offspring. So we're gonna say 2, this is the percentage. We will say 2 divided by the total number of the offspring. How many offsprings do we have? It's 1, 2, 3, and 4. So we have 4 offspring, right? So it's divided by 4. Because we are calculating a percentage times 100, and it gives us the total of 50%. percent. That's how we calculate the percentage for the offsprings, right? All right. Continuing, let's now go to the dihybrid inheritance. We have the dihybrid inheritance, which is the genetic cross between two individuals, which involves two characteristics. An example of a dihybrid cross, we have a purebred ram that was crossed with a purebred iwi. Now, we are required to use the Punnett square to determine the phenotypic ratio of the second filial generation, right? A purebred, meaning in terms of the genotype, it is homozygous. Both these animals are purebred, meaning they are both in a homozygous genotype. A ram, which is a male sheep, and also an iwi, which is a female sheep, right? The characteristics we're looking at the color, that is black, and also horned. The animal has horns or it does not have horns. So the first one, which is the ram, the male animal, it is black in color, and also it has the horns, meaning in terms of the genotype, when you are writing genotype, alleles are always written in pairs, right? So that is the genotype of a ram. When we are writing the genotype of a female, because it is recessive, we have the small letters, that is this recessive allele, and also another recessive allele. It is polled, meaning it does not have horns, right? Then we are about to use now a Punnett square. But before going to the Punnett square, for better understanding, we have to go through the genetic diagram first. Let's go through the genetic diagram before going for the Punnett square, right? We are asked to determine the phenotypic ratio of the F2 offspring, but we'll start with the first parent generation. The parent is the ram, which is a homozygous in genotype, and also another one is also in a homozygous state, which is an iwi. The genotype of the two, we have already said the genotype is the dominant alleles. The dominant alleles, those are for the ram. Then for an iwi, the recessive allele meiosis occurs, and then the gametes are formed. Then when the gametes are formed, each gamete has to contain an allele from both characteristics. For example, let's make it the first gamete. The first gamete will have an allele, a single allele from the genotype, a single allele representing one characteristic, that is a single allele representing the color, which is capital letter B, the dominant allele, and another dominant allele, which is H, representing the horned animal. This is the first gamete, and another one we also have the dominant allele. Coming to an iwi, it has the recessive allele for white, and also the recessive allele for polled. The recessive allele and another recessive allele. So fertilization will occur. When the fertilization occurs, we are crossing the male gamete and the female gamete, which will give us a heterozygous individual. A heterozygous individual and another heterozygous individual, right? Okay. That is the genotype of the first filial generation. The phenotype of these, they are all black and horned. Black and horned, because the genotype is heterozygous. The genotypes are heterozygous. So this is the phenotype of the first filial generation. Now, let's move. We are now going to the second parent generation. We are now going to the second parent generation, which is still the parent. Now, the offsprings are now the parents. They become the parent, the second parent generation, right? With the phenotype, they are all black and horned. So both of the parents are now black and horned, right? We have the genotype. The genotype we've already stated earlier that the genotype is heterozygous. Meiosis occurs, and then when after meiosis has occurred, gametes are formed. Then this time when the gametes are formed, each gamete has to contain all the characteristics. So all the characteristics must be represented. That is the dominant alleles. The dominant alleles, that is black and honed. One gamete will contain black and honed. Another one will contain black and poled. Another one white and honed, meaning all the 4 gametes will contain these characteristics. Same applies to the female, right? Then fertilization occurs where there's a fusion of the male gametes together with the female gametes, right? Okay. Then when it comes to the fertilization part, we now go to the Punnett square. We are required to go to the Punnett square. We are now going to the Punnett square. This is the Punnett square. When we are drawing the Punnett square for a dihybrid inheritance, when we are drawing, we draw a square. Remember, I said you just draw a square. We draw a square. This is a square for a dihybrid inheritance. A dihybrid cross. 4 lines down, 1, 2, 3, and 4. 4 lines across, 1, 2, 3, and 4. This is the correct Punnett square for a dihybrid inheritance. The first square, you can write gametes, or you can use our symbol. This is the female symbol. This is also the male symbol. Only use these ones when you are very sure that you know them. For example, let's say this side, the male gametes. This side, the female gametes. So you write the gametes. You write the gametes. These are the gametes for a male, capital letter B, and a dominant allele H, dominant allele, and a recessive allele, and a dominant allele. And lastly, they are both recessive. These ones are for the male. Then coming for the female gametes, we also have, again, by using these ones now, we have the capital letter, the dominant allele, and another dominant allele. A dominant allele black and the recessive allele poled. We also have the small letter that is recessive and honed, dominant. Lastly, we have the recessive and the recessive. So when you are crossing the two, when the fusion occurs, it will be a capital letter and a capital letter, and a capital letter. That is, it is the fusion between this gamete and the first gamete from the female side. And then the second gamete is crossed with the second gamete from the male. Now we have 2 dominant alleles for black, 2 recessive alleles for horned. You continue until you reach the last square. And then when it is complete, it looks like this. It looks like this when it is a complete dihybrid cross Punnett square. This is a complete dihybrid. So from the question, we were asked to determine the phenotypic ratio of the second filial generation. Right, we will write, we have the characteristics, black and honed. Black and honed, how many are they? Is 2, black and poled is 2. We also have number 3, we have white and honed. The last one, number 4, we have white and poled. Poled meaning it does not have horns. Okay, let's count now. How many are black and honed? This is 1, 2, The one that is heterozygous is black and horned. 3, 4, 5, 6, 7, 8, and 9. These ones are the black and horned individuals. Now let's continue to black and polled. Black and polled, it's 1, 2, and 3. Is 2, 3. Let's continue to the white and horned recessive alleles B and capital letter or dominant alleles H. We have 1, 2, and 3. That is 3. And then lastly, the last one is black and a polled. This is the last one, is 2, 1. That's how you determine the phenotypic ratio for the F2 offspring. All right, so now we have come to the end of our lesson. Don't forget to leave your questions and comments on the comment section. And also don't forget to leave your school name so that we'll be able to give the shout outs. Please share, recommend, and also subscribe to our channel. I thank you for watching, and I will see you on the next lesson, which will be on the patterns of inheritance. Thank you very much.